Now we'll see about the 4 marks answers, short answers in the zoology second year. In the short answers, lesson wise if you are seeing, as the digestive system lesson is not there, in the respiratory system if you are seeing, in the respiratory system, transport of carbon dioxide mainly consisting of 3 steps. The 3 steps mainly consisting of what? As dissolved state, water plus carbon dioxide gives rise to what? Carbonic acid. 7% of the carbon dioxide is transported in the dissolved state and 20 to 25% is transported in the form of the carbamino compounds. Whenever carbon dioxide is combining with the hemoglobin, then what we are getting? Amino hemoglobin we are getting, that is a carbamino hemoglobin we are getting, that is 20 to 25%. Reaction we have tried clearly. The third step we are saying particularly, that is actually formation of bicarbonate. Nearly 70% of the carbon dioxide is transported in the body in the form of the bicarbonate ions only. The first step which we had, water plus carbon dioxide gives rise to carbonic acid. The carbonic acid dissociates in the presence of enzyme carbonic anhydrase, and lastly what we are getting the bicarbonate ions. These are 3 steps by which the carbon dioxide is transported. If they want to ask for the 8 marks, particularly for the 8 marks, they are combining both oxygen plus carbon dioxide, they are asking for the 8 marks here. Oxygen transport is simply nearly 97% of the oxygen is transported with the help of the hemoglobin only in the form of the oxyhemoglobin, and 3% will be in the dissolved state we are saying particularly. It is one in the respiration. Other thing is actually mechanism of action of breathing, action of respiration particularly we are saying. It includes the inspiration and expiration. Inspiration is a simple phenomena of taking in of air particularly. In the inspiration, what is happening? Whenever the lung cavity size is increasing, air is entering. How the lung cavity size is increasing? Lung cavity size is always increasing by the contraction of the diaphragm muscles and external intercostal muscles. When these 2 muscles are contracted, what will happen? Lung cavity is increased. When the lung cavity is increased, what will happen? The air from outside is entering into the body. That is the inspiration phenomena. Expiration, it means what? Completely opposite to the inspiration. The relaxation of the diaphragm muscles and external intercostal muscles leads to what? Decrease in the lung cavity. When the decrease in the lung cavity takes place, what will happen? Air from the lung cavity goes outside, there is the expiration. Then respiratory volumes we are saying, tidal volume, inspiratory reservoir capacity, expiratory reservoir capacity, then residual volume, functional residual volume, and vital capacity. All these things we have to write in the respiratory volumes, that are also very important for the 4 marks. The definitions we have to give particularly, and then respiratory disorders. Common respiratory disorders we had, and then occupational respiratory disorders we had. Common respiratory disorders mainly consisting of pneumonia, asthma, emphysema, bronchitis. Pneumonia we are mainly by the Streptococcus pneumonia, respiratory breathing, problem in the respiratory breathing we are saying. Emphysema we had, alveolar walls are damaged in the emphysema condition. Bronchitis, inflammation of the bronchi walls. Then what we are saying, emphysema, bronchitis, all these are the respiratory disorders particularly we are saying. Then apart from that thing, occupational disorders particularly we had. If you are saying the occupational disorders, asbestosis, the persons who are working in the asbestosis industries, they are suffered with asbestosis. And silicosis, we are saying the persons who are working in the silica industries or sand industries, they are suffering with the silicosis. And siderosis, it means the persons who are working in the iron industries, iron mining industries, what will happen in the tissues, the small portion of the things will be jammed here, that leads to the siderosis. Black lung disease, the persons who are working in the coal mines, they are suffered by the black lung disease. These all the things are respiratory disorders. These are regarding the respiratory system lesson. After the respiration, if you are moving for the circulation, particularly the circulation differences between the arteries and veins, what are arteries, what are veins particularly, that we have to see. That means arteries are what, veins are what. Arteries are always carrying the oxygenated blood, veins are carrying the deoxygenated blood. How the artery size is, how the vein size is, what are the color of arteries, what are the color of veins, that things, all these things we have to write particularly. Then apart from this thing, cardiac cycle, sometimes they are asking the cardiac cycle for the 4 marks also. Then mechanism of the clotting of the blood, particularly extrinsic pathway, intrinsic pathway, blood clotting factors, what they are doing, that thing also we have to see particularly in the circulatory system. After the circulatory system, if you are moving for the excretory system, in the excretory system particularly, structure of kidney diagram is very important for the 4 marks. Cortex, medulla, columns of Bettini, renal pyramids, then renal artery, renal vein, hilum part, all these parts we have to see, and we have to draw a diagram compulsory for the 4 marks. And counter current mechanism. Counter current mechanism is a phenomena in which what is happening, loop of Henle, vasa recta, both are there. The loop of Henle, vasa recta which we had particularly, both when they are combining, by the filtration of the blood, what will happen? The concentration of the urine will be increased, particularly in the counter current mechanism. By what pressure particularly the filtrate is moving, that thing we are seeing in the counter current mechanism. Diagram we have to draw. We have to explain about 100 moles osmolarity. On the basis of the osmolarity we are giving the number that we have to explain in the counter current mechanism, that is in the excretory system. Diagram plus counter current mechanism we have to see, that is very important in the counter this thing, excretory system. If you are moving for the particularly in the neural control and coordination, diagram of the spinal cord is important for the 4 marks. Spinal cord diagram is very important. That is white matter, grey matter, then middle matter, that is actually head-shaped structure, central canal, dorsal horn, ventral horn. All these parts you have to indicate compulsory. Apart from this thing, synaptic transmission, electric synapse and then chemical synapse. What is synaptic transmission? How the synaptic transmission takes place? What happens in electrical synapse? What happens in the chemical synapse? Acetylcholine receptor, what is doing, and then different gamma aminobutyric acid, what it is doing. We have to explain about all these points in the synaptic transmission. Synapse is a point where ending of one neuron, starting of another neuron. That point we are saying as synapse. Synapse is a junction which is taking the impulse from one neuron and giving to the other neuron. That is synapse we are saying. Synaptic transmission includes the transfer of the impulse particularly. That we are seeing in the neural control and coordination, that is the brain. In the muscular system particularly, mechanism in the contraction of the muscle, axon and Hughes and axon, actually they are proposed with the sliding filament hypothesis. According to this sliding filament hypothesis, this is consisting of different steps here: excitation of the muscle, then formation of the cross bridges, power stroke, recovery stroke, relaxation of the muscle. These are the 5 steps particular. These 5 steps we have to explain, and diagrams we have to draw. What happens in the excitation of the muscle, what the calcium ions are doing in the excitation of the muscle, what the acetylcholine is doing, and in the power stroke, how the myosin head is attached to the how the myosin head is attached to the actin, how it is actually how that contraction, how the power stroke, what is the importance of the ATP, how the ATP energy is utilized by the actin particularly and myosin particularly to undergo the power stroke. And then recovery stroke, we are saying formation of cross bridges first. That means attachment of the actin to the myosin, formation of cross bridges. Next, power stroke, utilization of ATP. Recovery stroke, it means retraction of the particularly myosin heads from the actin. We are calling as relaxation of the muscle, particularly. And then all these points we are saying particularly in the muscle contraction. That points we have to explain. Diagrams we have to draw in the muscle contraction. Apart from this thing, if you are moving for the next, that is actually endocrine system. That is a hormones particularly in the endocrine system. Pituitary hormones, if you are seeing particularly pituitary hormones, growth hormone we had, prolactin we had, adrenocorticotropic hormone we had, then follicle stimulating hormone we had, luteinizing hormone we had. All these hormones which we had particularly about these hormones we have to explain. These are all the hormones of the anterior pituitary. Then median pituitary, if you are consisting melanocyte stimulating hormone we have to explain, and posterior pituitary if you are seeing vasopressin and oxytocin. Oxytocin we are calling as a birth hormone, vasopressin we are calling as a antidiuretic hormone. About all these hormones we have to explain the pituitary gland. And hypothyroidism, hyperthyroidism, secretion of the thyroxine hormone more we are calling as a hyperthyroidism. Exophthalmic goiter, that means the protrudence of the eyeballs we are calling main symptom of the exophthalmic goiter. Hypothyroidism is mainly explained by the cretinism and myxoedema. Cretinism always in the newborn babies, and then myxoedema we are seeing particularly in the adults. In the cretinism, mental retardation takes place, and then the growth of the baby is stopped, particularly in the cretinism phenomena. In the myxoedema condition, skin will become lethargy and mucus is jammed inside the body. There is a myxoedema condition. These 2 points we have to explain in the hypothyroidism. Then about the Addison's disease and then Cushing's syndrome. Addison's disease is actually particularly by the hyposecretion of the glucocorticoids. In Addison's disease, actually body becomes weaker, the metabolic activity of the body will be stopped, and the weight loss takes place. Mental retardation takes place mainly in the Addison's disease. In the Cushing syndrome, hypersecretion of the glucocorticoids lead to the Cushing syndrome. Moon face, buffalo hump, and pendular stomach is the main 3 symptoms of the Cushing syndrome we are saying. And the mechanism of action of hormones particularly. In the mechanism of action of hormones, membrane bound receptor mechanism, intracellular hormonal mechanism. Membrane bound, it means actually lipid insoluble hormones which are there, they are undergoing this mechanism. In this phenomena, epinephrine, if you are taking the example, epinephrine, if they are not crossing the lipid layer particularly, then what is happening? With the help of the membrane bound receptor, they are entering into the particular cell. In the cell, what is happening? Particularly G protein, which we had G protein. After G protein is actually converted into adenylate cyclase. Adenylate cyclase gives rise to what? CAMP is a secondary messenger, cyclic adenosine monophosphate. This secondary messenger gives rise to phosphorylase enzyme. This phosphorylase enzyme particularly we had, actually this gives rise to protein kinase A. Protein kinase A gives rise to phosphorylase enzyme. Phosphorylase enzyme is converting the glucose 6-phosphate into glucose here. This takes place in the lipid insoluble mechanism. Intracellular mechanism or lipid soluble, directly what is happening, the hormone which is entering into directly cell, in that thing it is entering into the nucleus. In the nucleus what is happening, it is converting into, directly giving rise to mRNA. Lastly mRNA gives rise to proteins in this phenomenon. This is the hormonal mechanism we are saying. After the hormonal mechanism, if you are moving for the immunity, that is the immune system. In the immune system, 3 defense line mechanisms particularly. First defense line mechanism with the skin, mucus and all these things. First defense line mechanism, natural killer cells, and then different cells. Second defense line mechanism. Then third defense line mechanism always consisting of lymphocytes and antibodies. If the antigen which is crossing this 3 defense line mechanism only, then we will get the disease. That defense line mechanism we have to see particularly. Then structure of immunoglobulin, very important. Actually structure of immunoglobulin is actually H-shaped structure. Paratope, it is actually antibody antigenic determinate site, actually it receives the antigen. Then H-shaped structure, light chains we had, heavy chains we had. Fragment antigen binding site upper side, fragment crystallized region lower side. We have draw diagrams and disulfide bonds. All these things we have to explain in the structure of immunoglobulin. And then about the different types of antibodies, IgA, IgG, IgD, IgM, IgE, all these things based on the heavy chains which we had particularly, we are dividing all these things here. Then about the different types of immunity, natural immunity we had, artificial immunity we had. Artificial immunity we are saying particularly, it is divided into active immunity and passive immunity. And active immunity again we have natural active, then artificial active. Passive immunity, natural passive, artificial passive, that we have to explain all these things. And in the immunity only particularly we have to explain about the mechanism of the HIV multiplication. In that phenomena, diagram of the HIV virus we have to draw. GP120 spikes, GP41 spike, lipid bilayer inside the SS protein, single stranded DNA, single stranded RNA particularly what is happening, how the transcriptase enzyme, reverse transcriptase, how it takes place, how the increase in the number of HIV takes place, all these things we have to see in the particularly immunity lesson particularly. And after, if you are going reproductive system actually, in the reproductive system, structure of ovary and structure of sperm we are seeing. The diagrams it is very important. And spermatogenesis, then formation of ovum, that is ovulation we have to see particularly. These are important in the reproductive system. And placenta, functions of placenta we are saying particularly, what is the importance of the placenta, all these things is very important. And then if you are moving for the reproductive health, the reproductive health actually different types of the STDs, gonorrhea, syphilis, Neisseria gonorrhea causes gonorrhea, and Treponema pallidum causes syphilis, and herpes simplex virus, human papilloma virus, genital warts, all these things. And then different types of STD disorders we have to see particularly, we have to explain about them. And then tubectomy, vasectomy, assisted reproductive technologies, about all these things we have to see, that is actually in vitro fertilization, and then gift and gift, gamete intrafallopian transfer, and then zygote intrafallopian transfer, then artificial insemination, and then all these things we have to explain in the assisted reproductive technologies. And all these things we had in the reproductive health here. Apart from the reproductive health, if you are moving for the genetics particularly, in the genetics, erythroblastosis fetalis, it is a phenomena in which what is happening here, if the mother is Rh negative, father is Rh positive, if the first baby is Rh positive, what will happen? The antibodies will be produced against the first baby, that is Rh positive baby. But when the first baby which we had particularly, up to the time but the baby delivery takes place, the antibodies will not be produced here. If the second baby is also Rh positive, what will happen? It is affecting the babies. The antibodies which are already secreted, they are affecting the baby here. That is the erythroblastosis fetalis we are saying. Hemolytic disorder in the newborn babies, the main symptom is a jaundice. There will be a chance of dying of the patient also. And sex determination also, sometimes they are asking for the 4 marks. Apart from this thing, in the genetics particularly, Human Genome Project is very important. That is actually, Human Genome Project is actually the United Nations energy system, particularly it has been started in the 1990 Human Genome Project. It has been ended in the 2003, 13 years it took for the completion of the Human Genome Project. Human Genome Project main aim is to sequence all the nitrogen bases which we are in the body, and to identify the genes which are there in the body, coding and non-coding sequences, to identify the sequence of the genes, and all these things these are actually present in the Human Genome Project target. But what happens in the Human Genome Project, what lastly they got, it means they have been sequenced nearly 4 billion, that means 4 billion nitrogen bases, that means 4 million nitrogen bases mainly they have been identified, and they have been lastly concluded actually 50% genes only they have been identified, and they found that actually only 2% of the genes which we had in the body, they are only coding the proteins particularly, and largest gene is a dystrophin gene in the body particularly. It is consisting of 2968 genes particularly, and the smallest chromosome is a Y chromosome, which is consisting of 231 chromosomes. All these points has been covered in the Human Genome Project. The main aim of the Human Genome Project is to sequence the human genes, and all these things is the main concept of the Human Genome Project. DNA fingerprinting technique. DNA fingerprinting technique is also very important particularly because for identification between the relation between the two persons, we are calling the DNA fingerprinting. The name DNA fingerprinting has been given because actually no two persons fingerprint will be similar. If the 99.9% similar, also 0.1% will be different. That's why DNA fingerprinting also, DNA sequences which we had, palindromic sequences if you are taking, restriction fragment length mechanisms if you are taking, no 2%, if the parents, mother and father also, children also, in them what is happening, DNA sequences will not be similar. We are saying in the DNA fingerprinting different steps we had: obtaining the DNA, cutting of the DNA, fragmentation of the DNA, separation of the DNA, blotting of the DNA, treating of the DNA with the probe, and all the things DNA fingerprinting we had. By the last DNA fingerprinting method, we are seeing the relationship between the person to person. Whenever the babies, whenever the crimes happen, whenever the scene of offense we are, they will collect the samples and they are doing the DNA fingerprint to identify the criminals particularly, to identify the culprits. And whenever the small babies they are losing from the parents, they are doing the DNA fingerprinting for confirming the parents particularly. This is about the genetics, important 4 marks answer. Apart from this thing, last applied biology, if you are saying the applied biology, animal breeding, inbreeding, outbreeding, close line breeding, offline breeding, all these things are very important. And then poultry management, dairy management, very important for the 4 marks. In the poultry management, actually what steps we are taking to increase the production of the poultry, boilers, layers. Boilers always grown for the chicken, and layers always grown for the laying of the eggs particularly. Then in the fishery management, capture fishery, inland fishery, importance of the fishery, what are the use, economic importance of the fishery, shagreen, cod liver oil, fish liver oil, then fish manure, fish guano, what is the importance of the fish guano, then importance of the honey, what from the honey what proteins we are getting, what vitamins we are getting, and honeycomb we are using in the polishing industry, and all these things we are seeing. And types of cancers particularly, carcinoma. Carcinoma is a type of cancer, particularly it is affecting the epithelial cells. 80% of the cancers we are saying as a carcinoma. Sarcoma is a type of cancers which are affecting the epithelial, which are affecting the tissues particularly. Sarcoma we are saying. Then next we are saying it as a leukemia. Leukemia is actually cancers which are affecting the blood cells, white blood cells mainly. Lymphoma are the cancers which are affecting the lymph glands particularly, or lymphatic vessels here. Types of cancers we have to explain, and what is the cancer. Improper, irregular, abnormal increase in the number of cells we are calling as a cancer particularly. Oncogens are the cancer causing cells we are saying. And vaccines, different type of vaccines. Edward Jenner coined the term first vaccine, whole attenuated vaccine, killer vaccine, and then toxoids we are saying. Whole attenuated vaccines, it means we are making the vaccines as inactivated, particularly mumps, measles, rubella vaccine we are calling as attenuated vaccines. Killer vaccines, particularly if you are saying we are completely killing the toxins, disease causing organism, that is the killer vaccines we are saying. Polio vaccines we are taking the example for the killer vaccines. Toxides, exotoxins, which we are actually exotoxins we are making inactivated, and we are making the toxins particularly. Generally toxins are using in the nowadays particularly. And then about the structure of insulin, insulin structure, the 3 chains which we are particularly, alpha chain, beta chain, particularly we are saying in the insulin. And then this all things are covering in the applied biology. In the applied biology also important questions which we are, they are very important. And MOET, multiple ovulation embryo transfer technology, is very important for the 4 marks. So this is also very important. And how we are producing the more number of particularly production in a less time. Multiple ovulation, that means many ovules we are producing by injecting the follicle stimulating hormone particularly. Embryo transfer technology, it means different in the different particularly parents we are growing the particular babies here, calves in a less period of time. They are the important 4 marks we are saying particularly. From every lesson we are getting the 4 marks. That means most of the 4 marks which we had in the zoology, particularly we have to write the important points. We have to cover all the points in the 4 marks answers. As choices are more, the nearly 14 out of 14 we have to write 6. Easily we can write the 4 marks answer, but we have to concentrate on the important. Apart from the important answer, we have to concentrate on the different answers which we had particularly in the phenomenon. Very important answers which we discussed now. Apart from this thing, you have to see the points of the 4 marks answers also. We have to see the points particularly.